Ever get tripped up by a passage describing a single base change and suddenly you're juggling missense, nonsense, and frameshift mutations in your head? And then the MCAT expects you to connect that to DNA repair pathways, like nothing happened. In this video, we're going to simplify it all, breaking down the major mutation types and the key repair mechanisms you actually need to know so you can read genetics passages with clarity and answer questions with confidence. Point mutations involve single base changes, such as substitutions, insertions, or deletions, which can alter protein function in predictable ways. A substitution replaces one nucleotide with another and can be silent, missense, or nonsense. Silent mutations do not change the amino acid due to redundancy in the genetic code. Missense mutations change one amino acid and may affect protein structure depending on the properties of the new residue. And nonsense mutations introduce a premature stop codon, often truncating the protein and severely impairing function. Insertions and deletions, when not in multiples of 3, cause frameshift mutations, shifting the reading frame and altering every downstream amino acid, typically producing non-functional proteins. A helpful mnemonic is CMINO for substitution types: silent, missense, nonsense. On the MCAT, expect to interpret how a mutation affects protein function, or to predict whether a mutation is more likely to be deleterious. Frameshift and nonsense mutations are usually the most severe. This concept ties closely to transcription and translation, especially codon reading and the importance of the reading frame, as well as to protein structure, where even a single amino acid change can disrupt folding or active sites. Chromosomal mutations involve large-scale changes such as deletions, duplications, inversions, and translocations that can affect multiple genes. A deletion removes a segment of DNA, often leading to loss of gene function, while duplication results in extra copies of a region, potentially increasing gene dosage. Inversions flip a segment of DNA within the chromosome, which can disrupt genes at the breakpoints or alter gene regulation. And translocations involve segment swapping between non-homologous chromosomes, sometimes creating fusion genes or misregulating expression. A classic example is the Philadelphia chromosome in chronic myelogenous leukemia, a translocation that produces an abnormal tyrosine kinase. For MCAT purposes, focus on how these structural changes impact gene expression and phenotype, rather than memorizing rare syndromes. These mutations connect to meiosis errors, particularly improper crossing over, and to genetic disorders that arise from altered gene dosage or disrupted gene sequences. Understanding these helps in passage-based questions where karyotypes or chromosomal diagrams are presented. DNA damage can arise from endogenous and exogenous sources, leading to mutations if not properly repaired. Endogenous sources include reactive oxygen species from cellular metabolism, which can oxidize bases, while exogenous sources include UV radiation, which causes thymine dimers, and chemical mutagens like alkylating agents that modify bases. These damages distort the DNA structure or alter base pairing, increasing the likelihood of replication errors. For example, thymine dimers create kinks in DNA that stall replication, while deamination can convert cytosine to uracil, leading to incorrect base pairing. On the MCAT, you may be asked to identify the source of damage or predict its consequence on replication fidelity. This topic connects to cell cycle checkpoints, especially the G1/S checkpoint, where DNA integrity is assessed before replication proceeds, and to cancer biology, where accumulated DNA damage can drive oncogenesis if repair mechanisms fail. Base excision repair and nucleotide excision repair are key mechanisms that correct specific types of DNA damage. Base excision repair target small, non-helix distorting lesions, such as deaminated or oxidized bases. A DNA glycosylase removes the damaged base, creating an abasic site, which is then processed by endonucleases, DNA polymerase, and ligase to restore the correct sequence. In contrast, nucleotide excision repair handles bulky, helix-distorting lesions like thymine dimers. A segment of the DNA strand containing the damage is removed, and the gap is filled in using the complementary strand as a template. A useful way to remember is that base excision is for small base fixes, while nucleotide excision is for nucleotide chunk removal. On the MCAT, expect scenarios involving UV damage or chemical exposure and be asked which repair pathway is involved. These mechanisms are also linked to genetic diseases such as xeroderma pigmentosum, where defective nucleotide excision repair leads to extreme UV sensitivity and increased skin cancer risk. Mismatch repair and double-strand break repair maintain genomic integrity during and after DNA replication. Mismatch repair corrects errors that escape DNA polymerase proofreading, such as incorrect base pairing or small insertion-deletion loops. It distinguishes the newly synthesized strand and replaces the incorrect segment, significantly reducing mutation rates. Double-strand breaks, which are particularly dangerous, are repaired by either homologous recombination or non-homologous end joining. Homologous recombination uses a sister chromatid as a template for accurate repair and occurs mainly in S and G2 phases, while non-homologous end joining directly ligates the broken ends and is more error-prone. A mnemonic is HR is high-resolution repair. NHEJ is not exact. On the MCAT, questions may test which repair pathway is active based on the cell cycle stage or the type of damage. This concept integrates with cell cycle regulation, especially the role of p53 in halting the cycle for repair or triggering apoptosis if damage is irreparable, linking DNA repair to cancer prevention.